Hey, it's Dr. Rosario, and I am a biology professor at Westchester University. Today, we're going to be covering a video on hearing, and in particular, the basilar membrane. So this structure is crucial and essential to our sense of hearing, but it can be a little bit hard to understand because of its coiled structure. And so I figured I'd go get a couple of images and we'd go figure it out. All right, so in terms of the basilar membrane, this is in the inner organ, right? The inner sensory organ of your ear, in the cochlea. And the one thing that you have to understand about this structure is it is really sensitive to the frequency of sound that you hear. This is actually what gives us our ability to hear different pitches of sound, whether that's like a really low pitch like bass rumble or a super high pitch like bee buzzing around. And so I'm going to show you a couple of images where we can see the basilar membrane sometimes coiled, sometimes really stretched out, and we'll see how that anatomy really relates to its physiology. All right, as nice as it is out here, it's a little too bright for me to show you some of the figures on my iPad, so I'm going to go pack it up. I'm going to go head back to my office, and then we'll pick it up from there. Okay, so we're back in the office, so let's talk about hair cells and the basilar membrane. Right, the first image that I want to show you, we'll just do a very, very quick review in terms of the structures of the ear. So here's your outer ear that is basically funneling sound to your tympanic membrane or your eardrum. The eardrum is going to vibrate back and forth depending on the pressure waves that make it to your ear. And that's going to cause these three tiny bones, known as the auditory ossicles, to vibrate back and forth and transmit that wave to the inner ear, finally making the stapes vibrate back and forth against this oval window. So we'll zoom in really quick over here. Here's your stapes. Here's your oval window. And a low pitch noise is going to vibrate the stapes back and forth at a slow frequency, back and forth like that. And a high pitch noise is going to vibrate the stapes rather quickly, right, with high frequency. Okay. So this figure I like because it has uncoiled the cochlea. So remember, typically the cochlea is going to look like a coil structure over here. This figure instead has uncoiled the cochlea so that we can focus on the basilar membrane. So the thing about the basilar membrane is it's going to bend in different locations based off of the frequency of sound that is being transmitted to the oval window. And what I mean by that is if you have this oval window being hit by the stapes from a low frequency, low pitch noise, that's very much going to bend the far region of the basilar membrane. So if you can see that, there's a little bit of an indentation right here. That part of the basilar membrane is going to bend in response to a low pitch sound. Right? Well, what happens if we hear a high-pitched noise? If we hear a high-pitched sound, we're going to vibrate this very, very quickly and instead bend the basilar membrane at this location. So this structure is going to be very important for our brain understanding, are we hearing a low-pitched noise or are we hearing a high-pitched noise? Okay. And the other thing about the basilar membrane that's very important to understand is embedded in the membrane, associated with the membrane, we have a bunch of hair cells. What do hair cells do? They turn bending into action potentials, right? Into signals that can make it to our brain. So when this part of the basilar membrane is going to bend because of a low-pitched noise, the hair cells over here are also going to bend, sending signals to our brain saying, we just heard a low-pitched noise, right? Whereas if we hear a noise over here, bending this part of the basilar membrane, the hair cells over here are going to make their way to our brain, sending action potential propagations to our brain, and our brain is going to interpret that as a high-pitched noise. Okay, so that's what's basically being shown down here. Here is a high-pitched noise of 20,000 hertz. If you're wondering what a 20,000 hertz noise sounds like, I'll just play it really quick. Okay, so it turns out that I might have chosen sounds that were really difficult to hear. So I'm actually going to show you a couple of tones that are a little bit easier to hear, and we'll play them in the video as well. So here's 200 hertz. It sounds pretty low, but you can kind of make it out if you listen closely. That's about 200 hertz. Let's bump it up a little bit. Let's see, what does 400 hertz sound like? Okay, a little bit higher of a tone. What about 800 hertz, right? Here I have 803 hertz. Let's listen. So as we go higher and higher to something like 2000 hertz, it's a much higher noise. Now I'm gonna play for you 20,000 hertz. I don't know if you're gonna be able to hear it because I can't, but maybe you will be able to. Here's what 20 hertz sounds like. Didn't even hear it. All right, so again, just wanted to give you some ranges of like what these sound like, what these tones sound like. All right, back to the video. So that's a 20,000 hertz sound that basically is going to cause your stapes to vibrate against this oval window 20,000 times per second, right? And again, that's going to trigger this part of the basilar membrane to send signals through the hair cells to your brain. So a 100 hertz tone is a vibrating your stapes on your tympanic membrane 100 times per second, which is actually going to cause maybe this region of your basilar membrane to bend, stimulating the hair cells that are embedded over here to send a signal to your brain saying, I just heard a sound that sounded like 100 hertz. Now, real sounds that aren't just pure tones are going to be a combination of many hertz. If you're hearing my voice right now, there are some low frequencies, some mid frequencies, and maybe even some high frequencies that are vibrating your eardrum at certain frequencies, and all of that is basically bending your basilar membrane in multiple places, sending that sound information to your brain. Okay, let's take a look at another figure. This one is a little bit harder to understand because we don't see the cochlea fully uncoiled, but hopefully you have enough information to really understand what's going on here. So again, here's the cochlea. You can see that it's folded, and we can still see sound waves making its way to the eardrum, vibrating the eardrum back and forth. And what this is trying to show you is if you have a high frequency noise, again, one that has a high pitch, it's going to bend one particular part of the basilar membrane. If you have a noise that's a little bit lower pitch, right, that's maybe going to, if I trace it back over here, maybe bend this particular region, which is going to trigger the hair cells that are over here, and then lower frequency noises are going to make its way further down over here and bend these hair cells. Right, now keep in mind that the basilar membrane and the cochlea are very coiled structures. They're going to be wrapped up and wrapped up and wrapped up in that kind of a coil-like structure, but really the basilar membrane acts the same way, right? If you hear a high frequency pitch, it's going to bend closer to the base. If you hear a low frequency pitch, it's going to bend farther away from the base. Okay. Now I just wanted to give you a little bit more detail because this figure does go into a little bit more detail. What if we zoom in on this box right here, right? What do we see? There are our hair cells, right? And the bottom of this is our basilar membrane that does all of that bending. Here are our hair cells that are sitting on top. And I'm simplifying the anatomy a little bit for you, but if you wanted to know a little bit more of what's going on, when the basilar membrane bends, that's going to cause the hair cells to push their hairs differently across this other structure known as the tectorial membrane, right? So what's really important is how do these hairs bend against this membrane? And as you can see, as the basilar membrane is going to bend, it's going to change the way that the actual stereocilia on these hair cells are touching the tectorial membrane, bending them that way. So there's a lot that's going on anatomically that I'm simplifying for you a whole bunch, but it's basically the same thing. When the basilar membrane bends, the hair cells associated with that part of the basilar membrane also going to bend. Okay, we have one last figure to show you, and this maintains the coil-like structure of the cochlea. All right, so what do I want you to focus on? Right here, this is the oval window. This is where the stapes is basically going to vibrate in. So if we have a stapes that looks like this, it's going to vibrate back and forth across that window, sending vibrations through this region towards the coiled part of the cochlea, right? The very end of it we call the apex, over here is the base. And what this is trying to show you is, over here, if we have a high frequency pitch, 1500 to 20,000 hertz, for example, that's going to cause bending of the basilar membrane very close to the base, maybe somewhere over here. If we have a medium frequency pitch, we're going to say between 600 and 1500 hertz, bend a little bit farther down. So maybe we're bending, let's see, 600 hertz is going to be close to here, 600 hertz, right? So we're farther down this coil, right? That's 600. If we're bending at 1500, we're a little bit closer to here. So if we did a 1500 hertz tone, we would be right here instead. But again, as you can see, the amount of times the tympanic membrane is vibrating back and forth is going to affect what part of the basilar membrane is going to bend. The most important thing I want you to understand, high-pitched noises are going to bend closer to the base. Lower pitch or lower frequency noises have to make their way all the way to the apex, right? They have to travel a lot farther down the cochlea to actually cause the basilar membrane to bend. So that's all I have for you in terms of the basilar membrane. Hopefully now you have a better understanding of how the anatomy and physiology of hearing works. How do we hear low-pitched noises versus high-pitched noises? If you have any other suggestions for future figures for me to go over, please let me know. And if you found this helpful, you can just do one thing for me. Keep going to class. All right, that's it for today. I'll see you next time.